certificate to Dr. Stroud. Also, please join us afterward for reception outside. And to those of you viewing this via live simulcast, welcome. I'm Elad Ziv, a member of the Committee on Research and professor of the Department of Medicine. Today it is my honor to introduce the Academic Senate Faculty Research Lecture. This award recognizes exceptional career achievement in basic science research. The Senate has two other faculty research lectures, one in clinical sciences in the fall and one in translational science in the spring. Today, we are proud to honor one of our colleagues, Dr. Stroud, professor of biochemistry and biophysics and pharmaceutical chemistry, with the 61st Faculty Research Lecture in Basic Science. Now I am pleased to introduce Dr. Oren Rosenberg, who will provide a formal introduction of Professor Stroud.
Thanks so much. Here's a slide to come up here. Thank you. I'm Oren Rosenberg. I'm an assistant professor in the Department of Medicine. I was a postdoc with Bob Stroud, and it's a huge honor for me to be here today to present this illustrious award and to join all of you. Bob has been at the forefront of structural biology since the beginning of his career. He was at the forefront when it looked like this, and he's still at the forefront now when it looks more like this, where we have single membrane proteins being gated by voltage in a membrane. And it's an amazing journey that he's going to tell you about today. But I wanted to highlight a few other ways in which Bob has had a huge impact here at UCSF. First of all, as a teacher, for the last 30 years, Bob has led the core biochemistry course, macromolecular interactions, and influenced countless students and brought and disseminated his unique style and rigor to labs all across the campus. He's had a huge impact as a colleague, primarily from my perspective. One of the most amazing things that Bob has done is spearheaded the macromolecular structures group, which has served as an intellectual home for structural biologists at UCSF, as well as a practical home with really critical cores, and even our own synchrotron beam line that Bob has spearheaded over the years and has turned into an incredible tool that's affected literally thousands of scientists. And it's an amazing accomplishment among many others. And finally, most importantly to me, Bob is an amazing mentor. And I'm sure all of you in this room have had the experience of interacting with him, and I know that his calm, humane, English accented voice brings immediate peace to many a hairy graduate student and faculty member. But I don't really feel like this introduction has done justice to the outsized contribution Bob has given to structural biology, and I've struggled with how we could work, to understand a little better what Bob has done. So I thought I would just run through some of the highlights of his 320 publications. So the first one was in 1969, Acta Christ, structure of.
Did you really want to do this?
Well, what do you want to do?
Do something different.
Peter, well.
I mean, come on, sing a song or something.
Well, okay. I mean, if you want to, we could do that. Yeah, sure.
How many years does it take to explore before Christmas grows through the cracks? How many ways must the black face one go before the spots can be tracked?
Everybody!
And how many postcards must one
Stroud.
No, that's another one. Oh, yeah. Well, that is such a hard act to follow. See Dave Eisenberg here as well. That's really beautiful. So thanks so much to the Academic Senate for this, for sponsoring this event. And thanks Oren and Peter and Dave for that wonderful undeserved introduction. It's just a tremendous honor to be here and to be listed alongside some of the greatest scientists in the world and the people I most admire and enjoy conversations with every day or so. This is just, I can't imagine anything finer than this. I want to acknowledge really every single person who joined our lab and carried out experiments, made discoveries, and allowed me to share in their discoveries. This has been really a true joy for me, and every single one of those people, I think, should feel that they share this honor with me. I think the award also recognizes structural biology. I think perhaps the first structural biologist to have been honored this way, and I just think of the outstanding community of structural biology here that we have among our faculty, and I'm just very proud of being associated with that group, with this group of wonderful structural biologists. So, let me start by seeing if the slides work. I'm assured that they do. So, title, Wiggle Wiggle Not a Trickle, which is connected to the movement of membrane proteins, structure, specificity, and integrity of biological membranes. This picture was generated by Julie Noodle, who for 30 years or so has been an illustrator artist. She's here with us in the audience somewhere. Where's Julie? You should all raise your hand if I mention your name. Brushwithscience.com. She's illustrated scientific articles for many of us faculty, Liz, Blackburn, John Sidat, many others. And this particular structure owes its origin to Dave Savage when he was a grad student in the lab, aquaporin, one of the aquaporins. And Dave is here, he's now a professor at Berkeley, and it's a great honor to see him here as well. Map of the event. I'm going to cover basically 4 numbered topics. We'll probably spend less time on each one going through. I want to begin by telling the current voltage sensor story that Oren introduced. I will not show the movie he showed, which is the gory details of what's going on, but I'll try and cover what I think is a new principle that emerges from this. And I'll talk a little bit about starting out in science. I got to say to all the students, if there are students in the audience, I think this is the most exciting time I've ever known in science. The possibilities are just enormous and beginning to open huge new horizons. I could wax on about that, but I think there's plenty of time at the wine and beer afterwards for that. Number 3, what have structures told us about membrane proteins? And then I'll cover rather quickly principles that have emerged from 4 different membrane proteins that we've studied. And then I want to end up on aspects of secondary transporters. And particularly in going through this, I want to acknowledge the sources of funding that have provided us with the ability to carry out experiments driven by discovery, driven by curiosity, through the UCSF sources, the PBBR, the Boyer Fund, and the Multi-Campus Research Program, and particularly the National Institute of Health that's funded all my research, still actually have the same grant that I started with on day 1 of my lab. It was still with us. And particularly I want to acknowledge Jean Chin, who has chaperoned me through the National Institute of Health for several years. She's now retired, so I'm, I think, allowed to show her picture and acknowledge her contributions to, really, to science and to being aware of what's going on and encouraging us. Why membranes? To me, membranes were always exciting because they come really before DNA and proteins, the separation of fluids between the harsh primordial soup and whatever chemistry goes on inside. They separate the chemistry, and yet they must then pass every single nutrient that comes into the cell, and also sense the richness of the environment, sense competitors, engage in biological warfare in the case of some of these bacterial toxins that we have studied, and elaborate signaling in eukaryotes. There are basically 2 kinds of proteins that are on the board here. There are also receptors, but channels that simply diffuse substrates, but specifically are mediated, and transporters that oscillate from one side to the other, driven by an energy source. So I'm sort of a structural biologist, though I'd like to think I'm a biologist too. And so what is structural biology? I think, to me, the idea was to understand how things work, and many of you, I think, would say the same thing. You want to understand how things work. Oh dear. Ryan, where are we? Authentication failed. This is the kind of thing which... Okay, I hope that doesn't happen too often. How things work. So, that means different things to different people. You could say, how does a car work? And I could say, well, you just take the key, put it in the ignition, turn it, and you can take yourself to work or to play or whatever you want to do. And that's perfectly fine. It gets you where you want to go. But if you want to know at a little bit deeper level, you might want a parts list. You might want a proteomic list of all the parts involved. That wouldn't tell you how they work together. And ultimately, the answer has to come from the dynamics of proteins, their multiple states, as Oren's movie showed, moving between multiple states to accomplish the function of proteins or membrane proteins. So at a level that connects biology quantitatively to the fundamental principles of physics and chemistry. And I think we all owe a great debt. You laid it at my feet, Oren, but I would lay it at James Holton's feet for having been the most creative person in synchrotron biology in the world by far. He's over there. And if you read his t-shirt, it says, "Never trust an atom, they make up everything." Fake news, right? They make up everything. So these are some of the membrane proteins that we have been involved in, not all of them actually, but several of them. Many from the plasma membrane, EPO receptor up here, and some from the endoplasmic reticulum. Here I see Peter watching over us, the homeostasis. And some in the endosomes. Now, much is known about the plasma membrane proteins. In particular, all of these proteins see an electrochemical gradient of 200,000 volts per centimeter. That's the gradient across the plasma membrane. All proteins respond in some small way to that voltage, but some of them use it, in neurobiology especially, to control the activity of proteins and sometimes channels. The channels are most well known. I'm going to focus on one particular protein as a means of trying to understand how voltage is sensed and how voltage changes the properties of a transmembrane protein ion conducting channel. And the reason why we chose an intracellular protein brought in from the plasma membrane in an endosome is because the richness of the control of the channel in the intracellular environment is so much greater than at the plasma membrane and offers us a way for the first time to see a voltage sensor in its resting state and its active state. So we can ask how it works at that level. So this project owes an awful lot to Alex Kinzer, who came from Berkeley, started as a postdoc in the lab, and here he is. Channels, typically, like Ron McKinnon's discoveries, are formed in a way that has a central channel that is ultimately gated in response to the action of voltage sensors that detect a change in voltage and relay it to a gate that opens or closes that channel. Opens or changes the probability of opening the channel. This TPC channel is found in the endosome, and it is there functioning in terms of trafficking of the endosome, ultimately for fusion trafficking of the endolysosome, which may fuse with the endosome or be recycled back to the plasma membrane. And look only at this one here, TPC1, that we're going to talk about. It is regulated, and I'm really only going to consider one kind of regulation. The fact that it is shut down by calcium, and calcium acts on the voltage sensing element. So we could use calcium then to switch this voltage sensor from its active state to its inactive state. And I stress that no one has seen, in a full channel, no one has seen a resting state even so far. Here is what the channel looks like, set in an endosomal membrane. Artist Janet Awasa drew this. An inhibitor here that actually cures mice of Ebola infection, which comes in through the endolysosome. So we know precisely which calcium ion sites are the ones that prevent the channel from opening by action on the voltage sensor device. So calcium we're going to use as a switch to switch between states. So Alex expressed and purified 17 species of these TPCs to obtain one that works, one that worked best, and as you heard, crystallizing is not always trivial. He then achieved what I would say is a heroic structure determination, 9 different heavy atoms, dehydration, many of them calcium mimetics, so that was 18 different heavy metal derivatives, as it were. And here you can see the central channel with some of those heavy metals bound that we use in a traditional way to discover the structures of proteins where you don't have an easy homologous model. So you're doing it de novo from the start. And once again, James Holton here telling us that yes, Schrodinger's cat is alive. He has really influenced us all by making the Berkeley light source, which we have a large part in the ownership of, a really great resource for students, especially students and postdocs, because of James himself. Now, the worst can sometimes happen, and it did to us in a sense, but maybe not. We found out about a couple of weeks before our paper was going to be published and was being somehow slowed down, we thought, at Nature, that there were two papers in the same issue of Nature, one being published by Yuxing Zhang's laboratory at UT Southwestern, structure of the voltage-gated two-pore channel TPC1 from Arabidopsis thaliana. And our title was remarkably similar, structure, inhibition, and regulation. So we had inhibition and regulation in here, TPC1 from Arabidopsis thaliana. We'd settled on the same protein from the same species. And Yuxing Zhang and Dejan Ren had added some beautiful electrophysiology that showed the channel opening responsive to voltage. We had some inhibition, the Ebola inhibitor, and many calcium binding sites, and so on that we had deduced. So what Dejan Ren and Yuxing Zhang showed was that as you bind calcium to this one site, and since it's a twofold symmetric molecule, there are two sites actually, there's a central channel in the middle, but it doesn't look quite like Rod McKinnon's structures because it's now only a twofold symmetric and not fourfold symmetric, and that's a sort of a detail. So calcium then changes the open probability from where it would be open at 0 millivolts, which is where all structural biology is done, at 0 millivolts, to a state where it would be closed. For the first time then, in a whole channel structure, in a resting state, closed in the presence of calcium. So the middle is a typical sort of channel, calcium, sodium channel, controlled by these very residues, which are what we call the selectivity filter. It's put together in a complicated way, but it's basically two units of the McKinnon-like KV channels in one chain, and it's wired in a manner, doesn't really matter, because the functional element in voltage sensing is due to the presence of 4 arginines in this helix 10. So I'm going to really focus the rest of this first thing on these 4 helices. These 4 helices are the voltage sensing element that detects voltage change and relays it to functioning of the pore in the center of the channel. So, voltage sensors have been known for 65 years since Hodgkin and Huxley, and discussed by Armstrong, Bezanier, McKinnon, and Hiller, and others. Basically, the phenomenon is this. If you change the voltage across a membrane, then you see a small current. You see a small current that goes away. The small current goes away because all it is charges that move from one side of the membrane to the other. And then if you take the voltage off again, those charges move back and you have a small current that you detect. And that change then in the voltage sensor is relayed to open and close the pore. These are some of the conventional ideas for how that process works. 4 arginine side chains, positively charged, move from one side of the membrane to the other. In the resting state, in a neuronal case, This would be, the positive charges would be down, and here they would go up. This was the notion of the conventional model, and McKinnon and Yuxing Zhang actually changed that and suggested that there was a paddling motion across the membrane that was what actually relayed itself to the central channel gating process. So the movement of one channel from the energized membrane, where there is a voltage across the membrane, typical voltage across the membrane of 60 millivolts or something like that, would change the way the energy profile, the free energy profile looks, to the point that the free energy associated with this arginine would move by 1.6 kilocalories per mole. I'm sure nobody really wants to know about that, but it's enough that in the TPC1, for example, the amount of energy is enough to change the open probability of the channel by a factor of 10 to the 5. And up here, the energy is much greater in the typical KV channel. So this is why all the neuronal channels seen at 0 voltage are in their active state, because a typical neuronal sodium channel, for example, opens in response to a 0 volts or voltage going diminished, depolarizing the membrane. And so these charges are, if you like to think of it, in the up state. But in the resting state, they would typically be in the down state with the 60 millivolts across the membrane. So this is our voltage sensor. Now, just focusing on those 4 helices at the top left corner, how does it work? How do they detect voltage change? Here are the positively charged arginines, 4 of them, locked together, bound together by a charge transfer center, an aromatic residue and a negative charge, that seal this voltage sensor to any leakage. Wiggle, wiggle, not the trickle, not the trickle. When we take a cross-section through this voltage sensor, you'll notice it's actually a little bit distinct from the pore itself, which would be over here. This is the 4-helix bundle voltage sensor. It appears in our resting state that there is solvent accessibility. So there's electrical connection to the cytoplasmic side, whereas in the typical case of activated state channels, the arginine's accessible to the other side, the outside of the molecule. So we have then a notion of how this could work. It could change the configuration of helices in a way which makes these charges detectable on the other side of the membrane. Could we find a way of actually seeing this happen? So to do that, we take Yuxing Zhang's result and remove the calcium, and then we should have a channel or a voltage sensor which is saying most of the time I am now going to be activated and open. So we wanted to see, this was the first resting state in any functional channel that's ever been seen, and we want to now see, could we activate it and see what happens. And the way Alex did this was by mutating this site, the effective calcium blocking site, mutating it away with these 3 mutants, and then we, now why am I not seeing, there's a slide missing here. Oh, here we go. Why did that get missing? Yeah, so to see if we could see this happen, we then teamed up with Evan Green and Yifan Cheng's laboratory, and his Alex and Pavel Dominik, who is a postdoc in the lab, and Tony Kozakov. A theme through this talk is going to be the continuity of influence of people. Tony was the first postdoc who joined me, as you'll see in a little bit, and we're publishing this paper with him again now. So that has been one of the luckier things for me in science, is the number of times that I've interacted again with the very folks who've made great discoveries in the past. And I think any electron microscopy can't go without mentioning Dave Agard, whose work with Peter Dennis, you may all be aware, converted the chip that Peter Dennis had come up with to a counter of electrons and made modern electron microscopy possible through the K2 detector. And Sasha Dickinson joined our lab this last year, and he's continuing this work with Evan now. Alex and Pavel have moved on to jobs. So the idea was then, use these mutants to switch states. We could see those states in these two-dimensional class averages that Evan Green was able to put together. And then he was able, very ingeniously, to assemble a three-dimensional reconstruction from the cryo-electron microscopy that showed precisely what happened to the voltage sensor, this colored element, that goes through a number of changes. And I tried to sketch it here last night to show what's going on. This is the resting state that we had before. These are the arginines, accessible to solvent from the cytoplasmic side of the membrane. And in the activated state, here they are, accessible on the other side of the membrane. So there's none of this movement up and down. It's a subtle twisting of helices which move around, in this case, to electrically connect arginines from one side, eliminate them, their access to the inside, and open their access to the outside. So that's represented here, and again I'm borrowing from Julie Newdoll's representation, who added the beautiful lightning bolts. So the bottom line here is that this voltage sensor at least, and we would like to know about others, of course, they're not all the same by any means, move by rotating around each other such that the arginines now connect electrically to the other side without any vertical movement. The voltage gradient is focused in the closing regions, and it's the voltage gradient drop which has really moved across the membrane, driven by that 1.6 kilocalories per mole per charge, 4 charges, 6.5 kilocalories per mole, that has driven the process and been able to then be harnessed to relate to the channel itself. So I think that reflects on the current possibilities of structural biology, particularly opened by the electron microscopy now, as well as the X-ray diffraction, which have really made it possible to see multiple dynamically accessible states in functioning molecules and deduce from that something about how they work. Okay, number 2. Twists along the way. This was perhaps the most formative experience of my life in science. I grew up in a little village near Manchester, England. This is myself and my brother. This was a garden shed. My father cut off 2 feet from the back, and he gave it to me when I was, I can't remember quite how old, but quite early on, and it became my first science lab. And I really used it. I had a transmitter across town to my buddy, and we had tape recorders. We designed and built them, and pH meter. I had a beaker of liquid mercury in there. That's why I'm in this condition now. So, oh dear, I keep jumping slides. Now then it's not moving at all. What's going on here? Brian, where are you? Uh-oh, they're frozen. Brian? Frozen. Well, I can give this slide while it's up anyway. One of my major influences at Cambridge as an undergraduate was this young man. The times they are changing. That came out in my last year at Cambridge. And I think this song, Only a Pawn in Their Game, is as poignant today as it was then in explaining the state we're in here.
You can sing it.
I could, for about 10 or 15 years of his writing songs, I could sing every one of his songs. And to some extent, I still do. I'm going to sing one later. If you listen carefully, but you might miss it. Oh dear. Well, I'll just keep talking on. So, from there, I went to Cambridge as an undergraduate, to Clare College, which is a wonderful environment to be in. Jim Watson was actually in Clare College, but I never knew that. And I would walk to work in the Cavendish Physics Laboratory, which was where we went for lectures. And every day I'd pass this hut on the right. I never really knew what it was. It was a hut, fair-sized hut. And it turned out that that hut was exactly the place where pioneering research was being done on hemoglobin, myoglobin, and DNA by Watson, Crick, and by Perutz, Ken Drew, and Richard Dickerson, who you'll hear a little bit more about if we do get any slides coming again.
The remote is confusing it, I think.
The remote, should I ignore the remote then?
Let's try it again.
Okay. Yes, here we are. So there's that hut. This is where they would win the Nobel Prizes in 1962 for work done here. And I think there's an underlying thought here, for me, that very often science gets conceived in ways that break the rules and break out of the mold of the way one thinks about things. And sometimes that needs, like this, environments that are borrowed from the cloisters of the academic ground underneath one. This is, of course, Willy Bragg's law. I came to know Willy Bragg a little bit later, not while I was an undergraduate though. And as I said before, my main influence was probably Bob Dylan. I indeed could sing every one of his songs. So then, after Cambridge, I looked around for a place to do a PhD. I had a last-minute switch of what I thought I wanted to do, and structural biology was really what fascinated me the most. And I went to work with J.D. Bernal. This was in 1964 for me. Some of his mentees were Dorothy Crutton-Hodgkin, also and Max Perutz, and Rosalind Franklin, Aaron Klug. Bernal was an amazing scientist. He had a fundamental paper on the structure of water. Dave Eisenberg told me about how this paper was written on a train, really, from Vladivostok to Moscow. Bernal was well liked in the East. And around the same period of 5 years in the 30s, Bernal and Dorothy had obtained the first X-ray diffraction from a protein, pepsin. And along the way, this was Bernal's camera. And it would work just as well today at the synchrotron, James, as some of our modern things. It has a slowly rotating crystal in the middle of a lead-cased thing with a film around it that would record X-ray diffraction from a beam coming in the side. So he was an amazing man. He was Churchill's right-hand man in science in the Normandy landing, so they were totally antipodal politically, but that's the way England is. And he helped plan those Normandy landings. I remember in my office one time I found this cardboard box of plates. There were stacks of glass plates, filmed glass plates, of the waves over the English Channel and planning the interference and how the Normandy landings would take place. He also wrote these several books that connect the social function of science, science in history, the origin of life, and so on. The first one is the most interesting, World, the Flesh and the Devil, inquiring into the future of three enemies of the rational soul. So once again, an inauspicious building was the key to some of the greatest science that was being done in that period by Dorothy and by Max Perutz and others. These houses, these were three houses in Torrington Place, bombed on both sides, that he begged from the demolition workers in order to build a viable crystallography department. And this was the front door. My office was up here, and he had his bedroom up there. So an occasional bedroom. This was a very memorable day for me. 1965, the Royal Institution, and this was David Phillips, and he just literally unveiled the first structure of an enzyme that was a structure of lysozyme. And here I was at the back, lucky enough to see this amazing thing. Here he had the unfolded protein. This was folds up and how it looked. And I remember Francis Crick was sitting right under here, and as he opened it up, he immediately pulled off the black cloth, and Francis Crick said, "Oh, it looks just like a clam. It's just like, where's the substrate? That must be good." And of course, he was right. So often was. Many times wrong, but okay. So I also learned and thought about and used one really guiding principle for me that we have used time and again throughout my career anyway. Structure determination, first of all, is totally independent of any assumptions. It is raw and therefore unbiased. But what these two gentlemen, Jerry Carl and Herb Hauptmann, had shown is that if you know something about an object, that knowledge can be integrated into the approach to the solution for it. Their mathematics was not honored for another 20 years from the time when we were using it, and it was then called direct methods. And if I distill it unfairly to the most simplest way I can say what they had done, was to take an assumption about an object and use that to help achieve the solution. The fact that electron density is always positive, and the fact that electron density could be approximated to some constant times electron density squared, which is what you see here. And that was what, now all of the small molecule crystallography is done this way, not the proteins, of course. But the principle is the key thing, that knowledge can be integrated. I'm going to mention that again. So I went to do a postdoc then. I'd actually been offered a job in Oxford with David Phillips, who had discovered the lysozyme structure. And he said, well, the post will not be available for 1 year. So if you could go and do a postdoc anywhere, somewhere, then come back and things will work out. So I went to work with Dickerson, Richard Dickerson, who had really done the mathematics and the computation, here he is with his paper tapes, outside that hut for the structure of myoglobin. The first structure of any protein was myoglobin in 1960-61, the very first protein. And he was at Caltech. Here he is. This is Dave Eisenberg, who you saw singing the song before. And I had no clue that he was coming here. Dave has been a wonderful colleague and friend over the years. We overlapped for a short time. These were the electron density contours that we would use to make models of proteins, and this was the way things were done. This was me at the time, and here we are again. The way we got these maps of densities was to contour computer printout and then translate it from computer printout into plexiglass sheets, and then use a mirror to build physically the structure of a protein to represent what it looked like. So these were hard times. It took a long, long time. Now a student can go and join James and in an hour come out with a structure if he knows, she knows what is going on. So I really appreciated the connection between art and illustration and what you're trying to say. And this came for me in the form of Irving Geis, who was renowned for his illustration of molecular structures, lysozyme, hemoglobin, and the like. Regarded as one of the greatest scientific artists of the 20th century, the HHMI website now hosts his work. And he illustrated this structure of the enzyme I had determined as a postdoc. That was the structure of trypsin and how it bound a substrate. This took him many weeks, and there were 13 overlays which made up this thing. I haven't represented it well in the figure here. And also represented the chemical reaction of an enzyme goes around and around and around, and is reversible, and so on. So he really made a big impact on me from the point of view of art. So now I mention artistic behavior again. I'm just going to begin here with item 3, what have structures told us about membrane proteins? So when I started my lab, I'd started with the structure of trypsin, and I'm going to treat it as a little wellspring up in the hills of insight. And I also was fascinated by membranes, and we started to work on acetylcholine receptor as a canonical membrane protein that was something we could work out. And as I started my lab, it was then at Caltech, I was unbelievably fortunate to have these wonderful 5 folks join the lab. Monty Krieger's down at MIT, Roger Kepi at Arkansas, Mike Ross, I'm not sure if he's here. Is he here in the audience? He may or may not be. He's just had a recent surgery. And Tony Kozikov, who was the first postdoc who joined us. So Mike took on very bravely the possibility of trying to understand what membrane proteins were like. And in this time, this is 15 years before the first membrane protein structure would be determined. So it was a long time ahead of its time in a way. And Mike built this camera specifically so that we could look at packed membranes and look at the diffraction and see what sort of structural information we could get out of membranes that were close packed with acetylcholine receptors in them. You needed big animals. Here's me cutting up a fish. There's a kilogram of electric organ here, which is richly endowed with this one membrane protein, well actually two of them, but that was the idea, and we purify the membranes from there. Toxins to purify, so you know what you've got. And this is the young Dave Agard, who joined soon afterwards, and Dave Agard and I spent a good bit of time developing methods of dealing with these continuous transforms. Mike Klimkowski took these electron micrograph pictures, which showed us some idea of how the density of protein in membrane was distributed across the bilayer. Here we are again. There's Dave Agard. You recognize him, I'm sure. He's up on the 4th floor now. This was myself in those days. Get a little Photoshop on that. And I was especially influenced by Mike Raftery. Mike Raftery was an amazing chemist, and it was he who opened up the field of neurobiology really by sequencing the 4 chains of proteins by Edmond degradation, amino acid sequencing, in perfection, along with Lee Hood, also at Caltech. He was also an amazing cook, not surprising for a terrific chemist as he was. So he was also an influence for us. Then one day, Bill Rudder, this would be, for me, probably the second most important event in my scientific life, was Bill Rudder offering me a position at UCSF, and I have never felt for a moment anything but the highest level of pride and honor in being here among the community that exists. When we came, John was here. John is over there somewhere, and Robert came soon afterwards. So we were doing structural biology. We were joined a few years later by Dave. He'd been a student with me and left a postdoc with John, and then a year in England. I don't know what he did in England. Anyway, came back. So here we are on the map. The acetylcholine receptor was the membrane line of thinking that was going on in the lab, and trypsin was enzyme creek. And enzyme creek quickly became drug discovery creek because, I would argue with everybody that we still do not understand one enzyme in terms of exactly how it achieves the high rate it does. Yes, you can borrow parts, mix them up, get an enzyme that kind of works, but you can't really understand it in the sense of knowing what it is that gives you that high rate of an enzyme. But knowing the enzyme structurally, you could design drugs that stop it working or that goose it up and make it work better. So on the acetylcholine receptor side, the membrane side, along the way, at UCSF now, Janet Feiner-Moore joined the lab in 1980. Here she is organizing the sequence of the acetylcholine receptor to come up with a prediction of how it would organize into the structure. Jörg Kistler in 1981 discovered these tubular crystals. Nigel Unwin did wonderful things with these tubular crystals. I think I could never have done it here. Would never have got continuing support from the National Institute of Health to study something and when it takes so long and is so difficult. So we focused on crystals, and Michael Schuster, who is here, where's Michael? There he is. Michael comes and gives a lecture here every year, a very important lecture for people who are thinking about industry and making companies, and getting them started, because he's now works as a partner in a law firm, particularly with patent law of biotech discoveries. And he focused on crystals of the acetylcholine receptor. We have these crystals that, as Oren, I think, alluded to, or the song alluded to, you can have crystals, and I'm going to allude to it again, that unfortunately don't ever give you the resolution to see the answer to the question that we really wanted to ask. So they are still important, and only now, this was 1990, the structure of the acetylcholine receptor was only described in 2016 for the first time from an X-ray crystal in its exact atomic way, and then it was only like 75% of the acetylcholine receptor. So these things are difficult. They're incredibly difficult because of the lipids in the membrane and so on. Same time, I should touch base with the other side of our interests. We're on enzymes. Dan Santi, I had connected with over his enzyme, a cancer target, thymidylate synthase. And by the time Janet joined the lab, she also worked on this, and by about 1984 or 5 or 6, I forget the year, she and Larry Hardy and Dan and I had been able to determine the structure of thymidylate synthase, and since then it's been the target for drug design experiments, some with Jim Wells and his group at Sunesis company, where we had papers together on using that enzyme as a proof of principle of the fragment discovery method. So just to underline the difficulties we were facing with thinking about membrane proteins at the time, this is 1960. The first soluble protein was discovered. Here is 1980, 20 years later, and the number of soluble proteins is rising like an exponent. 25 years later, the first membrane protein came along from Hartwig Michel, and since then, the exponent has gone up with a gradient which is years over 7 instead of years over 5, even slower, because of the barriers to getting insights into membrane proteins. Now, I think we're over this, but 25 years behind and an exponent, which reflects the difficulties that we were dealing with at the time. Another important protein in this period was not from the electric fish, but from bacterial sources, archaebacterial sources, bacteria rhodopsin. Bacteria rhodopsin, a very famous paper by Henderson and Unwin, looked at this protein and came up with an electron diffraction structure that showed that there were rods here. The rods seemed to be indicating there might be alpha helical regions that cross the membrane. It was still 20 more years before the detailed structure of this molecule was determined. Dave Agard, who was in the lab as a grad student, and I used that principle, the principle that if you know something, you can apply that in order to refine a solution to the problem. So we knew that the structure of Richard and Nigel had been limited because of the limited tilt angle of the electron microscope grids. So we realized that we could deconvolute this effect and get this structure. The reason these helices are straight is because it's 180 degrees away from that picture out there. So this was sort of the state of things. Here we are in 1982. In 1981, Stephen Hayward had joined the lab. He was a postdoc who'd been with Bob Glaser. Bob Glaser, who in my view is really the primary motivator for cryo, the cryo part of cryo-EM, Dubochet as well, but Bob Glaser really understood that the radiation damage was what was limiting and freezing he used to do it in a funny way, but in liquid nitrogen. And using his diffraction patterns, Steve came over to the lab, and we were able to phase these patterns to something like 3.7 angstroms resolution. So we knew, as other people knew, that membrane proteins had a structural integrity to them. And then we were joined by Larry Murky, an amazing guy who's here somewhere. Wave your hands, Larry. There he is. There he is. And Larry, when he came to the lab, he'd already crystallized in dim red lights in the dark bacteria rhodopsin, and he was also able to crystallize bovine rhodopsin, these big crystals, these huge crystals. And it was, again, it was 20 years ahead of when any crystal structure of bacteria rhodopsin was obtained. But colored proteins had a special meaning for us because they could be used to define detergents that didn't mess the protein up too much. And we even designed some new detergents along the way, and Ashish Mangalik, working with Brian Kabilka, has also been involved at Stanford in developing others. So this was the group. Mary Batlack is here, over there. Where's Mary? Yeah. And she cloned the gene for bacteroidopsin, and we were able to express it. Alok Mitra did some of the electron microscopy as a postdoc, and George Turner worked with Mary on expressing bacteroidopsin. So we were really pushing the limits to try and get as far as we could. Now, over the other side of the river, I got to say I chose this analogy for life in science because it seems to me that the discoveries that you make along the way, and the ideas and the concepts that enter the stream, never leave, and they really get reinforced with time and come back again and again to have impact in the way we think about things. Over on this enzyme drug design route, there was Jim Hurley. He was a biophysics student in our lab, and he's now a professor. Where's Jim? He's here. Yeah. Thank you for coming, everybody that has come, and Jim especially, and Jim and Dave Savage. And he was working on phosphorregulation up here at the time. We also got involved in HIV proteins. We had a long collaboration with Charlie Craig, 15 publications at least. And on HIV integrase, HIV protease and integrase, two of the only three enzymes in HIV, and these structures have been used many times to assist in drug discovery for Gilead Company and others that have designed drugs to inhibit, for example, integrase and conjugated with protease drugs. Andy Levitt, who was going to be here, but Eli Levitt is sitting in his seat, which I have named, and I want everybody to know that seat had Andy Levitt's name on it, because he asked me to have a front seat named, and then he didn't show up, so. And Harold. Again, Akram and Julian Chen and Janet Feinamore again, who is, of course, still a key player in our lab in so many ways. She just worked on the enzyme that tailors the primer for HIV from lysine 3 tRNA and showed how that works. And it was really Dan's idea originally that if we inhibited that enzyme, we could prevent the priming of HIV, and it has about a twofold effect. Now, on the other side of the other side, in the membrane protein river, Mike Ross was here again. He had a company, involved me in the company a little bit. We worked on the EPO receptor. That led to a nice publication on that complex, and much work with Jim Wells, who'd done the HGH receptor complex before, how to understand cytokines. I'm going to stop in these two islands, water channels and gas channels, and just touch base on what we learned along the way. So here was the first membrane protein structure in 1985. Here was the first ion channel from Rob McKinnon in 1998. Here we are in 2000 with a description of the first water channel, and here was the first gas channel up here in this island. So water channels, what about water channels? Humans have 12 water channels. In the course of this lecture, there will be something like 2,000 liters of water going through your water channels to reabsorb water from your urine, so you don't have to run out there and release it. So you're saving water. And then in the highlands and other places are aquaporins. Now the interesting question about aquaporins is they're kind of all built the same. They have a continuous line of water through them. And that's important because water goes through water channels at the diffusion limit for a hole of this size. That itself is amazing. Diffusion limit. They go around as tetrameric assemblies, 4 water channels like this viewed from on top. This is a side view, water's in the way. So how is it that they do not leak? Because, wiggle, wiggle, not a trickle. There is no ion ever goes through a water channel. No ion of any kind, and no hydronium ion. No proton even. Most of these water channels are very similar. Here's Dave again, one of them. Here's Larry, who's a part of many of these analyses. They all kind of look the same. How could they eliminate the leakage of a proton? Protons are known to tunnel through lines of water molecules and appear on the other end by just this quick switch around. So no oxygen has to move from place to place. How does that happen? The way it happens is because the line of water molecules is, first of all, sequestered from solvation, so a hydronium ion with a plus sign on here and an extra proton would have a difficulty getting in the first place because of the water of hydration, which is a key. But also in the middle of the water channels are 2 hydrogen bonds that orient this central water so that all the other waters point north and south. They're polarized. So a proton coming in could never get to the other side. So they have eliminated the leakage of protons by that clever mechanism. What about the gas channels? The gas channel is in the RH factor, and we've looked at a number of RH proteins. The latest one in process right now. They are channels that conduct ammonia. And how do they do so without leaking, say, potassium ions? Potassium ions have the same ionic radius as ammonium ions, and they do so, and the structure instructed us into this. I'm showing no mechanistic assays, though in every case we carried them out with liposomes, with proteins incorporated. So the way they do it is the ion comes in, is stripped of its proton to leave ammonia. Ammonia goes to the other side to be rehydrated, take up a proton, and detected by a pH change on the other side. So this was regarded highly by chemists, at least. It was one of the 4 highlights of the year. And we knew it was important because a few weeks later, here was Carl Robinov, a geologist, published in Science magazine, seen reading the issue of Science with our cover on the surface. So we knew it was an important discovery back then. Now there's another river that I haven't mentioned up here. This is to do with membrane protein biogenesis by SRP, the system that Peter Walter discovered with Gunther Blobel. and we had an amazing time working on this over many years. This is 1983 when Peter arrived, and we still talk about it and are working on related things. Along the way, Pascal Legier worked on the complex that seals the deal between the recognition particle and the receptor that talks to the translocon, where the nascent membrane protein or secreted protein would be inserted. And he discovered the structure of one of the, not the first one, but the third translocon, but one that told us a little bit how it opens into the membrane. And here again is Peter. We've had a long time collaboration, 17 publications together. This is the UPR IRE1 FLUSA. And this is where we worked with Peter on proteins from the system he discovered there. On the other side, Son, Nadine, Janet, Joe worked on this HIV primer problem. And down in the membrane side, near where I'm going to finish up in a moment, Sergey and Oren work on these proteins. We're working with Oren on trying to, now this is bad. So we're trying to understand the membrane proteins of TB, Mycobacterium tuberculosis, on the grounds that there is at least one of them we know is essential. So the idea would be then, could we inhibit that one with a drug? Sergey joined the lab a year ago. There have been some stars in our former life in the lab. Thomas was the most recent one who left, who's championed ABC transporters in our lab, and here Bjorn and Drew have now started their own operations elsewhere. But item 4, the finishing item, is really how do secondary transporters work? And really, so many of us have contributed. This is the current lab today, and many of them, if not all of them, have contributed to this. We've had a collaboration with Ron Kabak, with Hemant and Janet contributing there. And the latest news you will hear soon from our collaboration with Rob Edwards, and I will generalize about all of these experiences. So forgive me for not showing any real data here. Coming to what I hope will be an important and an interesting conclusion. What are secondary transporters? At the level of the ignition key in the car, as knowing what they do, you could be very happy to know that the way they really work is by taking a proton or a driving ion of some kind, coupling it to a substrate, and moving it across the transporter, however that may work. Biology doesn't like that mechanism very much, but that is the net effect of how these secondary transporters work. They work by wiggle from one side to the other, never leaking any phony substrate or non-substrate, because they are specific, but they bind very weakly in order to be able to give up their substrate on the other side. The only time a proton is involved, I'll say it now but show it in a minute, is when they recycle empty to the other side. Otherwise, the proton is here all the time, or the driving ion is there all the time, as it moves from side to side, without leaking, without binding too tightly to block it, and yet being highly specific to just the transport of the substrate. So, how could this be important? Here is an example. Chiadi and Janet determined the structure of the glucose transporter, particularly the one that is augmented in many kinds of cancer cells to feed the cancer cells the 9-fold excess of glucose that they need to function. And this is an inhibitor. This is the first structure of this kind of a transporter with an inhibitor in there that is a putative drug as a means of blocking this particular GLUT1 glucose transporter. So I think there is then some possible future in understanding these structures at this level, a level which begins to close the gap to what we saw on the enzyme side. So here's a lot of words. Let me just say, I sort of said they bind weakly, they do not bind tightly. You know, some people think, even my colleagues, some people think the way they work is they grab the substrate on one side and they pull it in and push it into the high concentration against the gradient. And so they can concentrate, substrates 10,000-fold. So the way they actually work is like this, and this summarizes a lot of work and a lot of thoughts. If you had a closed system, imagine a closed system now. Don't worry if you're not thinking like a chemist, it doesn't matter. We'll soon get it visually displayed. So there is a free energy difference between one side and the other. This is Boltzmann statistics, which reflects the difference in concentration. If there was a leak here, the protons, which are the energy source of a bacterium or a mitochondria, would be leaked away. Or the sodium ions would be leaked away in a eukaryotic cell. So there's no leak. If you put substrates on one side, and the transporter works by taking substrate and proton together to the other side, they will move to a new equilibrium. And the new equilibrium will have energy both equivalent on both sides. The ratio of concentration 1/2 will be 2/1. And that'll be a stable state then, oscillating backwards and forwards, but not changing concentrations anymore. So this is basically how they work. And what we've learned from working with Ron Kabak especially, in the beginning, but now several transporters in our own lab, is that the binding of substrate occurs by one pathway. The binding of protons occurs by a different pathway in space and in time. And that's what is the bottom line here of how biology solves this problem of how the car works, how the machine works. So let me just, with this, oh dear. Oh, may as well just jump to the movie then. No, the sound is off. Is it on? Oh yeah. So here's the transporter. This is a titratable group. Titratable group. This is the outside of the cell. Some highly energetic protons moving around. Some specific substrates are out here, the hexagons. On the inside, could be the inside of the cell, mitochondria, or it could be inside of a liposome. So inside, there are already some substrate molecules that are concentrated here. Here they are. And there are some protons energetically moving around like that. A little bit of water back there. Secondary transporters concentrate substrate using proton symport. So this is a symporter in the same direction. There's our proton stuck on the negative charged residue in there. Substrate specifically binds hydrogen bonds. Can go from inside to the outside, be detected now on the outside. No change in proton movement from one side to the other. and another substrate can go back. This is called exchange. Another substrate molecule can go back. But nothing would change the concentration yet, because one comes in, one goes out. Here we are again. Water is a key player in all of this. It's a new substrate. Yeah, she fits the site, got the right action bonds. She can move to the other side and appear on the outside of the cell again, on the outside of the lipocell. So, we can prove, or Ron Kabak first proved to my knowledge, that substrates can move through these transporters by using radioactive substrate, even when the proton site is inactivated. So no proton-driven force, no secondary ion-driven force. They still exchange, detected by radioactive substrate. They wiggle, they can take specific substrates from one side to the other. Now non-specific substrates, they may not have an arm or a hydrogen bond, right? So they get excluded by just having zero binding, or no binding at all. How does the proton drive this event? How does it make that marriage with the substrate? It only makes it when the transporter closes and goes back empty. When it goes back empty, the pK drops and the proton leaves. And of course, as soon as it reaches the other side, a proton will immediately lock onto that now raised pK again. So every time that now we've got business as usual, substrate can bind again, substrate will go from outside to inside, protonation doesn't change. Once again, then, the site is filled with water, and if it goes back to get another bunch of substrate, the proton is driven vectorially. That's a real question. How does the vectoring occur in the right direction? So now we got the last demonstration here that I think as the transporter closes to go back empty, a proton is driven out from the site in a vector direction. and Fong, who put that together from a single take. So that, I think, is how we think transporters work. We know it in a couple of cases, and I think Rob Edwards and I are going to describe a very interesting case between our groups coming up. And let's see what else. I think I am truly honored to have worked with the people on that acknowledgement shrine. I can imagine no higher honor than to be here sharing this moment with you. Thank you all very, very much for coming here.
I just want to say on behalf of the Academic Senate, we're truly honored to give you the Faculty Research Lecture Award in Basic Science in recognition of your outstanding achievements in basic science.
Well, thank you very much.
And then I think we should soon transport ourselves outside. There'll be refreshments and more talking for foot.
Good to see you. Remember Pablo Valenzuela?
It's been a long time. Yes, of course.
Anyone stopping there?
We can let it go.
Erase everything.
Yeah. That's the problem.
I'll have to tell them that I read it too. How are you? Good to see you too. You know Pablo, right? Pablo Valenzuela? It's so good to see you now. I've seen you.